Hello, everyone. Myself and Dr. Arna Christensen have been investigating the influence of microplastic color and odor on feeding preference in Daphnia. And Daphnia are freshwater crustaceans. You can see a close-up video of a Daphnia here, as well as some Daphnia feeding on some microplastics on the left. Just to give you some perspective, Daphnia are about the size of a grain of rice, so they aren't quite microscopic, but they are quite small. I also mentioned we are working with microplastics. And these are small pieces of plastic that are less than 5 millimeters in diameter, and microplastics are found everywhere. But the most common type of microplastics in aquatic ecosystems are microfibers, and those are the types of microplastics that we use for our experiments. One of the concerns with microplastics is that small organisms like Daphnia are eating them, and as these small organisms eat microplastics, there are larger concerns of bioaccumulation and biomagnification, as bigger organisms eat small organisms like Daphnia, allowing microplastics to move up the food chain. So microplastics are being directly consumed by these small organisms, but still making their way into top-level predators, including us. For our experiments, the first thing we needed to do was determine how we could analyze these microplastics. We were initially worried about determining fiber color just from these bright field photos in this first column here, because the colors of the fibers get washed out a bit for our green and our blue. The black and the red fibers were able to be identified with these bright field photos, but it turned out that our colored microplastics have unique autofluorescent properties. So you can see that our colored microplastics here all autofluoresce red, but the key was that our green microplastics autofluoresced green, and this allowed us to distinguish our green microplastics from blue. And this was great news because it gave us a mechanism to identify fibers with a high degree of confidence. So having a way to analyze our future data, we set off to conduct some experiments. For our experiments, I needed to prepare microplastics by finely cutting up some polyester fabric and making stock solutions of microplastics in combo media, which you see here. We also had a constant culture of Daphnia, so we had Daphnia on hand when we needed adults for experiments. But before Daphnia were exposed to microplastics, they were fasted for 24 hours to make sure they would feed on microplastics and their intestines weren't too opaque. And then we had two main sets of experiments, one looking at microplastic color and one looking at odor, which were done independently. But for both experiments, Daphnia were exposed to microplastics, then the intestines were dissected and imaged, the fibers were counted, and our data was analyzed using those autofluorescent properties that I talked about. Our first set of experiments looked at color preference, and my hypothesis was that Daphnia would prefer green microplastic because they would resemble their algal food source. So we set up 3 treatments with a 50-50 ratio of green microplastics mixed with either black, blue, or red. And here you can see some Daphnia feeding on the microplastics in an exposure container. And each of these treatments contained 8 animals that were fasted for 24 hours and then exposed to a final concentration of 0.01 milligrams per milliliter of microplastics for 1 hour. And after exposure, these animals were fixed, dissected, and imaged. And here you can see one representative image of the dissected intestines for one animal for each of these groups. And when we went through and analyzed this data, we didn't find a significant preference for color in any of the groups. There was a slight preference for black and for blue in these first 2 groups here, and a slight preference for green in our red and green group. However, this data was not statistically significant. But things got a little bit more interesting as we moved into our odor experiments. This involved aging microplastics with algae to biofoul it. So we took some green and some black microplastic and we aged it in combo media with algae or without algae for 3 weeks in our greenhouse, which you can see over here. Those are our 2 groups. And the microplastic aged with algae is referred to as fouled, and our microplastic aged without algae is referred to as pristine. And my hypothesis here was that the Daphnia would prefer microplastics that have been biofouled with algae because they would mimic the olfactory cues that algae normally secretes. So we used 4 different treatments here with a mix of these green and black biofouled or pristine plastics. And again, each treatment contained 8 animals that were fasted for 24 hours and then exposed to a final concentration of 0.01 milligrams per milliliter of microplastics for 1 hour. Then the Daphnia were fixed, dissected, and imaged. And here you can see an image of the dissected intestines, one representative animal from each of these groups. So we have our pristine microplastics on the right, a mix of fouled and pristine in the middle, and then our foul green and foul black microplastics here on the right. And because we were just using green and black fibers, we focused on the green emission in order to count our green fibers and our bright field to count are black microplastics. When we went through and analyzed these microplastic counts, we see a clear preference for black across the board. And this difference is significant within all of the groups with biofouled plastics, which you see here. And when looking at the ratio data across groups, it's clear that the preference for black is heightened significantly in the presence of biofouled microplastics. The thought here is that it isn't actually the biofouled microplastics that are more attractive, but the fact that there are olfactory cues in the water that are promoting feeding behavior in the Daphnia, and that's heightening this underlying preference for black that otherwise isn't significant. The preference for black is most intense when both sets of microplastics were biofouled, so we believe that the more olfactory cues in the water there are, the more heightened this preference for black will be. Lastly, if it was the individual plastic pieces that were more attractive, we would see a preference for green microplastics in this group here, where we have fouled green microplastics and pristine black, but that was not the case, as there's still a preference for black here, indicating that it's the presence of these olfactory cues in the water that's triggering this feeding behavior and heightening the preference for black microplastics. If we were able to continue our research, we want to confirm that these olfactory cues are in fact diffusing into the water from our microplastics. We want to identify which chemicals are being secreted into the water by algae and determine the threshold for these olfactory cues to impact preference. Lastly, it would be interesting to determine if our findings are true for other types of microplastics besides the microfibers that we used. Lastly, we would like to take a moment to thank Dr. Christensen, as well as Dr. Stevens and Dr. Giuliano for all of their support throughout this research project, and thank you for listening and learning a bit about the world of microplastics and Daphnia with me.